Hi, everyone. Welcome to today's webinar. All right. Hi, we're going to leave it open another minute to make sure everybody has time to join. Nice to see so many people are connecting. So, as I said, hi everyone, and welcome to this third episode of the spring mini-series on restoring across depth. This webinar series is brought to you by Ocean Citizen Project. It's a Horizon Europe project about restoring marine forests. So just so you know a little bit more about the project, it takes place in 5 different ecozones. One of them is Tenerife in the Canary Islands. And why am I mentioning Tenerife? Because the speakers of each of the 4 episodes of this series are actually leads of the restoration of one type of marine forest here in Tenerife. Tenerife is the main site of the Ocean Citizen Project. The objective is obviously would love to tell you we are restoring Tenerife's marine forest. But the objective is a little bit more modest than that. It's to develop blueprints, so robust protocols for the restoration of different marine forests across depth. So we have intertidal, shallow, mesophotic, and even deeper interventions. And the goal is for these protocols to be replicable and scalable. So this is also the reason why it's taking place in 5 different places. For episode 3 today, we have Gerard Mas Jimenez, who's connected now from the CEAB, working together with Rafael Coma on the restoration of Gorgonians. And Gerard will introduce to us basically of all the keys, all the different concepts to take into account when developing a project to restore Gorgonians. We're going to wait another minute for everybody to join. In the meantime, you can maybe drop in the chat where you're joining from. I am based in Tenerife, in the Canary Islands. And yes, before I forget, remember this episode is being recorded. So it will be made available through our YouTube channel and website in just a few days. The episodes 1 and 2 are already available on the YouTube channel, just so you know. So if anyone missed something, I can tell you that you can catch up without any problem. Okay, still some people joining. Before I leave the word to Gerard, I just wanted you to know the presentation is going to be roughly 20 minutes. After that, we're going to move to a quiz that we have prepared, like in the last episodes. In case it's not your first, you already know about that. And then after that, we will circle back to the Q&A, to which both Gerard and Rafael are going to be attending your questions. Just to make it easier for us to manage questions and make sure not a single one is left unattended, please make sure you put them in the Q&A box and not in the chat. It's going to be easier to find them afterwards. So that is it. It's 4:05. I think we can get started. Gerard, it's all yours.
Okay, thank you. Moment. Sorry. Okay. Hello, my name is Gerard Mas. I am currently doing my PhD thesis in the field of demography, population dynamics, and restoration techniques, working on Gorgonian forest. First of all, I would like to thank Isabel and all the people involved in this series of webinars for giving me the opportunity to present my research within the European project Ocean Citizen. Today I will introduce the general topic of this talk, entitled Restoring Gorgonian Forests from Science to Success. In general, when we think about restoration, we think of something that has been damaged or disturbed. However, the full framework of the causes of damage and the future scenarios is not always well known. Therefore, before making any decision, it is essential to shed some light on this complexity. First, I would like to refer to the Society for Ecological Restoration, which provides a widely accepted definition of ecological restoration, shown here. That is as the process of assisting the recovery of an ecosystem that has been degraded, damaged, or destroyed. Today, this definition is broadly accepted by the scientific community. It is important to understand that an ecosystem that has been degraded, damaged, or destroyed becomes a different system, with multiple possible trajectories. One possibility is the natural recovery, which occurs when stressors or impacts disappear. Other states may persist while coping with the impacts, resulting in a different but functional ecosystem. If mitigation actions are applied, ecosystems may recover partially or totally. However, when mitigation is insufficient due to severe damage, compensation measures may be needed, such as creating alternative functional habitats. And finally, restoration can involve an active process, developing techniques and actions to reinforce the recovery of the original ecosystem. Then, this means that the ecological restoration aims to reach the highest possible level of recovery based on the reference ecosystem or a reference model. This reference model must be clearly defined using all previously gathered knowledge. In this picture, you can see a reference model, a healthy ecosystem, and then a damaged ecosystem that is damaged, and maybe we can decide to restore. The objective of the restoration is to recover the ecosystem's capacity to function and evolve naturally, either following known recovery trajectories or adapting to global change. And then this is the possibilities and the possibilities of the restoration according to the feasibility of making the restoration available. For example, in the best condition, if the site conditions are still broadly suitable and the impacts are mitigated, then we can use the full recovery ecosystem, the reference model in the high level. When not, maybe you will have other possibilities depending on the feasibility of them. And at the end, if the restoration is very hard to make, then you have to select the most restorative option, trying to restore some of the functions that have in the past this ecosystem. After defining the reference ecosystem, the first step is to assess the level of degradation. This includes evaluating the biodiversity through indicators such as species mortality, invasions, sublethal effects, and reductions in the reproduction and growth, as well as identifying the causes of this mortality or this damage to the species. In addition, it is necessary to assess how the ecosystem degradation affects human health and well-being. For example, through harmful algal blooms, declines in fisheries, or the loss of valuable seascapes. The second point, all the stakeholders involved must understand the full picture, addressing past and present conditions and the causes of the impacts, such as the temperature increases or ocean acidification. Also, we have to know the socioeconomic factors. The legal frameworks and cultural activities must also be considered to understand the whole picture of the ecosystem in the present. Therefore, it is crucial to compile all the important factors, the baseline conditions, and the pressures that the ecosystem have to make a whole picture of the ecosystem right now, and then analyze what kind of restoration we can do. Once the ecosystem is well understood, restoration planning and design can begin. This includes defining appropriate restoration actions based on local conditions, as well as setting clear goals and objectives. The planning should anticipate failed activities and, importantly, incorporate an active management approach. This is very important because during the process of the restoration, maybe there will be pros and cons of doing restoration actions, and we can adapt the restoration to this knowledge that we achieve during this process. Then adaptive planning allows the integration of this experience gained during the process, including both successes and mistakes. With a well-developed plan, restoration can be implemented. Then we have another step, that is the implementation. Ensuring that benefits and resources are equitable distributed among stakeholders. Without stakeholder support, conflicts may arise and jeopardize the project continuity. Then it's very important to go together in a way to make the restoration feasible at the long term. During this implementation, pilot or preliminary tests are essential to evaluate feasibility and success. Here we have images of some kinds of restoration process, and then with analyzing these experiments, we can gather important knowledge about how it works, or if it works or not. Then then it's important to collect all the data that we made during these experiments, recording the methods, the actions, and the outcomes, because this will be important to allow replication in other ecosystems or regions, or try to improve these techniques to maximize the outcomes of the restoration we are doing. Also, recording all the methods and techniques will increase the value and the impact of the project. And then it's after doing this implementation, these actions, and developing the restoration actions, then we have the ongoing management, monitoring, and evaluation process. The restoration is a long-term process rather than a single action with immediate results. Therefore, monitoring and evaluation plans are essential to assess project performance and improve future restoration efforts. The monitoring must be repeatable, involve stakeholders, and be based on objective criteria. In this regard, the Society for Ecological Restoration provides useful visual tools to assess restoration success now and in the future, as you can see in this picture. Then the definition and measurement of restoration success require flexibility and alignment with local needs. Careful selection of success criteria is necessary, as some benefits may otherwise be undervalued or overlooked. According to objective criteria, restoration outcomes can range from pressure reduction, we will be here, or to full ecological recovery. That will be at this point. With higher levels, these levels of recovery of natural ecosystem representing the greater restoration success, the higher level of restoration. Well, after providing this context about restoration and the whole process, I will now focus on our work within the Ocean Citizen Project. Specifically, we work on the Gorgonian forest restoration. First, we assess the reference ecosystem that was a Gorgonian forest in Gran Canaria, in the Canary Islands, next to Tenerife, dominated by two species, the red and yellow Gorgonians. This forest extends over a large and relatively flat area. We assess physical parameters such as the temperature, the nutrients, the substrate composition, as well as the pressures affecting the conservation of this Gorgonian forest. We also defined restoration goals, including where and how to restore Gorgonian forests, how many colonies to transplant, how to obtain fragments without damaging the natural populations. That is very important in a restoration. Based on this information, we implemented restoration actions and first we conducted 3 preliminary experiments. First, we assessed the thermotolerance of these two species. Then we assess the better transplant support for the fragments of the Gorgonians, and also we make nurseries in aquaria and in the field to increase the growth of the fragments. That is essential to improve the survival of the fragments in the restoration site. Regarding the physical environment, we assess the annual thermal regime. Under natural conditions, the seawater temperature at 25 meters approximately ranged between 18 and 24 degrees during the year. However, in 2023, we recorded temperatures above 25 degrees Celsius for the first time at this site, at Gran Canaria Island, and also in Tenerife, in the restoration site. The two thermal regimes, the one of the natural population in Gran Canaria and the restoration site in Tenerife, more or less match the temperature, the thermal regime. This temperature recorded in 2023 was an anomaly that can threaten Gorgonians, and for this reason, it must be considered during the restoration process. Then we also assess the nutrient concentrations during the warm season. Concentrations of essential nutrients such as particulate organic carbon and particulate organic nitrogen recorded in the natural habitat confirmed to us the oligotrophic conditions that are characteristic of the Atlantic Ocean. Then, in terms of physical environment, we assess the substrate occupation, and we observed that 92% of the seabed consisted of rocky substrate, providing suitable habitat for organisms such as gorgonians, sponges, and algae. These results give us information such as the place of the restoration site must have substrate characteristic like this, to allow the Gorgonians colonize naturally the restoration site, with the sexual reproduction that is the natural process of colonizing. Then we study the key species, the Gorgonians, in the natural habitat, in the reference ecosystem. We studied the demographic parameters of the Gorgonians, including the conservation status and the size structure. Both the species showed low partial mortality, less than 10%. That that is considered as a good conservation status. However, in the study of size structure, we detected a low abundance of small size colonies, suggesting limited recruitment or high early mortality, or both. Although this result, we must think that these species are long-life species that can reach 100 of years, and this study is about 3 years. Then the scale is not the same, and we would need long-term studies in the future to assess in a better way the recruitment and and the mortality of these species. Then, after that, we assess the growth and mortality of the red Gorgonian in this case, and we saw that the growth is approximately 1 centimeter per year, more or less, which can be described as a slow growth species. Also, we saw that the mortality rate was higher in the small colonies, in the colonies with less than 10 centimeters in height. Potentially this is affecting the recruitment consistent with the size structure observations. And the results show us that the first stages of the Gorgonians could be hard to surpass and affects the survival of them. And the final study we made in the sense of the assess the reference ecosystem in terms of the key species was the study of the sexual reproduction, which is the main characteristic to develop healthy populations with diverse genetic Gorgonians that can cope with present and future perturbations. In this regard, we observed that the colonies tend to reach sexual maturity at the small sizes, with colonies larger than 5 centimeters generally being mature. And here we have an image that shows gonads, specifically spermaries, in a male colony fragment. So some of the results of this assessment are still in study. Then, in terms of the implementation of restoration action, we made the experiment of the thermotolerance under controlled conditions. And here, if you remember, in 2023, we reached seawater temperatures above 25 degrees Celsius that can affect the survival of the Gorgonians. Then we studied the thermotolerance of this species because we think that this is an important factor before transplanting fragments in another place, which can take temperatures like that. But our results showed that the first signs of necrosis were not observed until more than 30 days. 30 days at a temperature of 25 degrees Celsius. That is more or less here in both species, in the red and the yellow Gorgonian. And this time, these 30 or more days, are 6 times more days than the recorded in 2023. That we recorded more or less 5 days this temperature above 25 degrees. Then this result tells us that the Gorgonians won't suffer mass mortality due to the increase of temperature alone. This is very important because we saw mortalities when the temperature was above 25 degrees Celsius in the Mediterranean red Gorgonians. Here is the Mediterranean red Gorgonians, that when we have less days at 25 degrees Celsius, the Gorgonians begin to die, and they are very severely affected during the past 10 decades. This is a picture of a Paramuricea clavata, a Mediterranean red Gorgonian, with dead parts recovered by other organisms after these parts of the tissue were necrosed. But this is not the case for the Canary Gorgonians, which have higher resistance, include temperatures above the 25 degrees that are the maximum recorded in Tenerife and as well in the natural habitat in Gran Canaria. Then we assess the transplant support of this, trying to improve the growth of the fragments. These experiments are in the aquaria. First in the aquaria. As you can see, these species grow very slowly, and we aim to enhance their growth, collecting small fragments, and then in the aquaria trying to overfeeding them to maximize the growth. At the same time, we try to improve their survival by testing different supports, optimizing the attachment of the fragments to the support, and overfeeding them. We try to make the best conditions for the colonies to generate the holdfast that will contribute to improve their survival in the long term, because in the natural habitat there are currents, there are very hard currents. Then the other experiment we made was trying to deploy floating reefs that will contribute as nurseries to make also more or less the same as we made in the aquaria, trying to collect small fragments of the of the Gorgonians, elevating them 1 meter above the substrate to reduce sediment abrasion and competition with other organisms that grow fastly, such as algae. And in this regard, we also noticed that that there are pros and cons of transplanting fragments depending on the size of them. For example, in small fragments, during the collection of the fragments, the damage to the donor colonies is less than if you collect larger fragments. But when you collect small fragments, then there are some cons that affect the survival of these fragments, like sand abrasion, the detachment of these small fragments, the predation exposure, the sexual maturity as well, because they're required to grow to reach the sexual maturity. And for this, these experiments aim to improve the growth rate of small fragments for reaching this size enough to survive better, and reach the sexual maturity that will increase the natural colonization of the rocky habitats in the restoration site. Well, the results of these preliminary experiments, by integrating these results and identifying the critical points, we developed objective conclusions and restoration strategies based on the most effective techniques. This will ensure the long-term sustainability. And at this point, we will transplant. We want, and we expect that we can, that we will transplant the fragments obtained of these experiments to these kind of structures, the cell structures, that will serve as a substrate for the Gorgonians and a refuge for other organisms. And as I mentioned before, after implementation the restoration, the monitoring and evaluation plan will allow us to study the biophysical conditions at the restored site, trying to detect anomalies in the thermal regime or something that can occur, and to monitor the demographic parameters and population dynamics of the transplants. This will help us to understand where we currently stand and how the system may evolve in the future. Always remembering that the restoration is a continuum, a process that must be adaptive and collaborative. And finally, in the sense of broad engagement and sharing, the goal of Ocean Citizen project is to serve as a pilot study, generating transferable knowledge for future restoration efforts, pointing out the steps that must be done in order to improve the success of the restoration. And this dissemination of these results is key to maximizing the impact and the value of the project that could support further research and restoration initiatives. And that's all. Thank you very much for your attention. I hope you found this presentation interesting. I will be happy to answer any questions you may have in the next minutes. Thank you very much.
Thank you, Gerard. That was great. So we do have a couple questions in the Q&A box, but as I said earlier, I suggest we go first to the quiz. So I'm going to share the link for everybody to join the quiz. I'm sharing this in the chat, right? There you go. So you should be able to join the quiz from that link, all of you. And on my side, I am gonna share my screen if the computer lets me. Share my screen. Yes, there it is. So you should be able to see that. Is it all good on your side? Yes. Oh, let's jump into question 2. Okay, sorry. So let's get started with the quiz. I'm assuming everybody could get in. Yeah. So first question, which environmental factor is especially important for Gorgonian forests because it directly influences their feeding? So we have 4 options, A, B, C, and G. And it seems like we already have a preferred one. So indeed it is B, water currents, which transport plankton and organic particles. And that is what the Gorgonian feed on. Moving on to the next question. Why is understanding Gorgonian population dynamics essential for restoration planning? Again, 4 possible answers, A, B, C, and D. Leaving you to it. Is it to estimate how quickly colonies can recolonize new areas? Is it to determine how populations grow, reproduce, and respond to disturbances? Or to identify which colonies produce the most oxygen? Or is it rather to measure how fast colonies can attach to artificial structures? And that's interesting. We have a 50-50, 40-60. I'm unsure whether everybody had a chance to answer already, but we have 2 candidates here, A and B. So part of the audience is answering with A, to estimate how quickly colonies can recolonize new areas through normal dispersal. And the rest of the audience is going for B, to determine how population grow, reproduce, and respond to disturbances. So I don't know if either Gerard or Rafael, you want to jump in and comment on why is it B and not A or both?
Isabel, I think there is in the chat that it seems that that the presentation has frozen.
Okay. So I'm going to stop sharing for a moment. Presentation is frozen, but you 2 can still follow me, right?
Yeah.
So there's a mystery. Okay. Can the rest of the audience hear me well and see? Well, I'm stopped sharing my screen, but can you all see us? Yes. Okay, so apparently part of the audience can still see and hear us. Maybe it was just momentaneous little glitch.
Maybe. Okay.
I'm gonna go back to sharing the quiz then. Hopefully it will go well this time. And we were busy with, okay, question 2. Start presentation, full screen. So is it good? No one commenting that they cannot see the screen?
Well, I think that's the correct answer, and it's critical. I mean, to understand the demographics of the population in order to see whether the restoration site is successing, is working properly in order to approach that scale that Gerard has shown, that is evolving towards a natural population. So, I mean, it's a good answer.
All right. Thank you.
You're welcome.
Thank you, Rafael. So we're going to move to the next question. Please let us know in the chat if there's any issue. Question 3. In the context of Gorgonian restoration, what does assessing thermotolerance help determine? So is it how colonies respond to changes in light intensity? Is it B, how resistant colonies are to sedimentation events? Is it C, how well colonies can withstand elevated temperatures without stress or mortality? Or D, how quickly colonies can grow in deeper waters? I do think this one was an easy one. So we have 100% of you answering C, how well colonies can tolerate elevated temperatures. Question 4.
Isabel,
Yeah.
Isabel, I think the presentation is frozen.
Again?
Yeah,
Oh.
I think.
Well, you know what? Yeah, I'm not sure what I can do.
Well, it doesn't mind if you say the results, that's fine.
But yeah, I believe, so obviously I'm on the presentation side, but once you join with the link, the assistant should be able to see the question and the possible answer on their screen. So we'll just move forward and let's see how it comes out in the recorded video. So fourth question was, what is the main purpose of growing Gorgonian colonies in nurseries or aquaria before transplantation? We have 4 options, and all of you answered B, to increase colony size and strength. And this is my bad, because the right answer is B, to increase colony size and strength before exposure to natural conditions. And there seems to have been an error when I put the questions in the automated quiz, because indeed it is answer B. So when you go live, you never know what's gonna go wrong.
Okay.
And last question for all of you, 5, why might restoration projects combine methods such as aquaria, floating nurseries, and direct transplantation? Again, 4 possible answers. Is it to reduce the need for environmental monitoring? Is it to standardize restoration across all habitats? Is it to optimize survival and growth under different environmental conditions? Or is it rather the last option, to accelerate reproduction cycles artificially? So the audience, the public is quite cohesive on that one. Let's see if I put the right answer in the system. Yes, I did. So it's answer C, to optimize survival and growth under different environmental conditions. So thank you. Oh, we have another answer. Someone replied B, to standardize restoration across all habitats. Well, I don't think there's much to go over to correct that answer B, because it's clearly to make sure every fragment you're gonna collect is gonna survive. We never have 100% survival rate, I think. I don't know if I can say never, but I'm pretty confident about that. Maybe you can confirm, Gerard or Rafael. But indeed, using so many different methods, it's to make sure that everything we collected has the highest chances of survival.
Well, Gerard can confirm it, but in the aquarium, the attachment of the substrate to the substrates of the fragments has almost reached 100%. Is then in the field when you have the currents, the fireworms, I mean, all these other actors that can reduce, but in the aquarium, I think from more than 700 transplantations, I think that only one failed, no, Gerard?
Yeah. Yes. For this is important, very important to to try to improve the generation of the holdfast of the Gorgonian attached to the substrate to improve the survival in the field, in the natural habitat, or in this case in the restoration site, because then the currents in the first stages of the restoration, it's very easy to detach the fragments, because the attachment of the Gorgonian to the substrate is very is not hard enough to sustain the current of the water movement in the natural conditions.
Okay, thank you.
Isabel,
Thank you
if
for
you want, I
specifying.
can answer the first answer.
Yeah, I was
question.
gonna say, so I'm gonna stop sharing my screen, and we're gonna go to the Q&A. I don't know, Gerard, if you can put the slide, just so we have.
Yeah, I will try.
So with regards to the first question on
yes,
whether
thank you, Rafael.
how did we increase the temperature in the thermotolerance experiment, we did increase following the natural increase rate of temperature in the field that we had previously recorded. And each one of the treatments did increase up to this rate, up to the level of the treatment. Like, for example, if we started at 20 degrees, when you reach 24, that treatment will remain constant over there. Another treatment will continue increasing up to the 25.5 and will remain over there until we see a signal or a problem with the colonies. And the same with the next treatment, which will be the 27, keep increasing up to there and remain at that constant temperature until the end. And then the same with the 28.5 degrees treatment up to the end. And the amazing result was that the colonies resist for a long time period all these temperatures.
Thank you, Rafael. So we have another question here in the Q&A box, and it is, how did you obtain the fragments? Did you tear them from the reference point or use some broken ones?
Do you want to answer, Gerard?
Yes, we collect the fragments from the natural habitat in Gran Canaria. We try to collect small fragments from the donor colony, from natural colonies, and we take only one fragment of about between 3 or 5 centimeters long. One fragment from one colony to not damage a lot the donor colonies.
So if just to add a little bit to your answer to that question, as Gerard mentioned during his presentation, it's like the donor colonies are being, obviously for studying the reference point, ecologically speaking, I guess you're taking fragments from different colonies in different stages or health states. But then once you go for the restoration action implementation, you take targeted colonies that you know have the characteristic and features that will enhance the success of the restoration. I don't know if either of you can maybe mention, because I think it might be interesting, that such projects do exist that collect broken pieces, loose fragments, maybe through the fisheries, and then replant them with a method that is called badminton method, which is not what we're doing here in Ocean Citizen, but I think it's worth mentioning.
Yeah.
I don't know who wants to go first. So this other kind of project is where you're focusing on putting back those pieces that got broken and taken out of the water accidentally. And this Ocean Citizen Actions is the focus is different. It's rather to find the right method to have an effective and successful restoration.
Okay, we see another question about how did we measure the growth of the colonies. This has been monitored, well, and is being monitored by means of taking pictures of the fragments at regular periods with a scale.
Yeah.
With this, we measure increase in size, but also in the area of the fragments, which allows us to estimate variation in biomass.
Thank you. I don't know if there's any other questions from the public. If you still have some doubts or some curiosity about Gorgonian restoration, just go for it. This is your time. In the meantime, I'm already going to share with you in the chat because it looks like oh, we have another question in the Q&A. I like that. Great. Can you see it, Rafael, or should I read it?
Which one? The one from Marta regarding the success of restoration? This one?
Yes.
Well, no, I think that this method, the method that is being used by the team from the University of Barcelona, the broken fragments, I think that is doing quite well. It's much difficult to know the success because they come afterwards with this is used usually for much deeper use. I mean, they just get from the fisherman and pull it back quickly to the sea. So it's difficult to assess efficiency because they do not come back after for about a year. But I think it's quite efficient, and they can use it. The good thing about this is they can use it with larger Gorgonians, and I think it's really efficient.
So it's maybe these two methods are quite hard to compare, right? Because the monitoring is really not the same. Like your techniques include a strong monitoring plan post transplantation, and the badminton method doesn't have such a strong monitoring plan. But it also, I guess, the effort investment initially is also quite lower with the badminton method than it is with like a thoughtful restoration like you two are leading. Thank you. Okay, no more questions? It looks like we have answered all questions. So as I was saying, I'm gonna drop in the chat the link to our next webinar. It's gonna be next week, so next Tuesday, same time. You can register through the link I just shared on the chat in case you haven't yet. Next week, well, at first it was going to be a 3-episode miniseries, and in the end we have a bonus episode, which is this one, number 4, and we'll cover restoration in the mesophotic zone. So mesophotic is this transition area where there's still light, but just not as much as there is in what we know as the epiphotic zone, or the first 30-40 meters. And this will be brought to you by Francisco Otero and Lorenzo Bramanti, two researchers that are also part of Ocean Citizen Consortium. And well, please join. I'm sure we're going to learn a lot about restoring black corals at such great depth. Well, thank you for joining and see you soon online. Bye. Thank you, Zora. Thank you, Rafael.
Thank you.
Thank you.
Thank you very much.
Thanks.
Thank you.